Hello everyone, a warm welcome in today's lecture. Today we are going to study the chapter cell division, and in cell division we are going to study in detail about mitosis. Now we know that life of all multicellular organism, it starts from a single cell, and this single cell, when it divides, it results in growth of living organisms. So it has been seen that in living organisms, two types of cell division are seen. One is mitosis and other is meiosis. So let us start in detail about mitosis. Now when we see mitosis, it is a type of cell division in which a cell divides to form two similar daughter cells, which are identical to the parent cell. As we know that a single cell is formed initially, and that single cell then later it divides. So we can say mitosis, it is a type of cell division in which a single cell divides to form two similar daughter cells. That means from a single cell, two daughter similar cells are formed, and these two daughter similar cells, they are identical to the parent cell. Now it is completed in two steps, that is karyokinesis and cytokinesis. Karyokinesis, it is a nuclear division which is subdivided into prophase, metaphase, anaphase and telophase. Now we know that every cell, it has cytoplasm and it also has a nucleus. So when the division takes place in the nucleus of the cell, that is called as karyokinesis. So we can say karyokinesis, it is a nuclear division. Whereas cytokinesis, cytokinesis, when mitosis, it is a continuous process that starts with disappearance of nuclear membrane in prophase and ends with separation of two fully formed cytokinesis. So mitosis, it occurs in two stages. First is karyokinesis, where division of nucleus takes place. And when the division of nucleus is completed, cytokinesis, that is division of cytoplasm takes place. Now when we see karyokinesis, it has four substages. The first stage is prophase, second is metaphase, third anaphase, and last is telophase. So as here it is said that after mitosis, after the division of karyokinesis, cytokinesis takes place. So let us go first in detail about prophase. Now when we see prophase, it is the first stage of mitosis. This phase involves condensation of chromatin material, migration of centrosomes, appearance of mitotic apparatus, and disappearance of nuclear membrane. Now here are the two cells. Yes, okay, let's see. This is the cell. This is the cytoplasm of the cell, and this is the nucleus of the cell. Now when we see prophase, prophase is divided into early prophase and late prophase. Now what happens in early prophase? Now basically when we see prophase, it involves condensation of chromatin material. Chromatin material basically it deals with chromosomes. Initially when we see in early prophase, the chromosomes are in the form of long thin thread like structure, okay, which is also called as chromatin material. So the chromosomes you can see, they are in the form of long thin thread like structure, okay. And then migration of centrosomes. Now what exactly is centrosomes? Besides the nucleus, you can see centrosomes. Centrosomes, it always gives rise to centrioles. Okay, two centrioles are formed from a single centrosome. So these are centrioles, and centrioles, they are always formed from the centrosome. Then appearance of mitotic apparatus. Now mitotic apparatus, it refers to formation of spindle fibers. Okay, so mitotic apparatus means what? Spindle fibers. So what basically happens in prophase? This involves condensation of chromatin material. Condensation, the meaning of condensation is not what the chromatin material, it reorganizes itself to form a short and thick structure. Okay, so the meaning of condensation is the organization, the reorganization of chromatin material. ये जो पूरा chromatin material है, this all chromatin material, it reorganizes itself in such a way that they become short and thick. See, in early prophase, you can see they are in the form of long thin thread-like structure, and when they undergo condensation, you can see here they become short and they become thick, and that is why they have named here as condensing chromosomes. Now, due to condensation, each chromosome becomes visible under light microscope, which can be seen with its sister chromatids connected by a centromere. Now, as we had seen that initially the chromatin material is in the form of thin thread-like structure, but in late prophase, this was the case in early prophase. In early prophase, also nucleolus, you can see it is very distinct. Whereas nuclear envelope, okay, nuclear envelope, it is a protective covering around the nucleus, which is called as nuclear envelope. So in early prophase, prophase can be divided into early prophase and late prophase. In early prophase, you can see nucleolus is distinct, nuclear envelope is very much distinct, as well as the chromatin material is in the form of long, thin thread-like structure. But as the early prophase progresses, what happens? The chromosomes undergo condensation. And as they undergo condensation, when we see under light microscope, each chromosomes, they become very much visible, and in them you can see sister chromatids connected by centromere. Now sister chromatids means what? The chromatids of the same chromosome. Further, I'll show you what is sister chromatids. Okay, you can see this is a single chromosome. Each chromosomes, they have sister chromatids. This is one and this is two. These are the sister chromatids. So chromatids of the same chromosomes, they are called as sister chromatids. And the structure which holds the chromatids, this is called as centromere, which is situated in the center. Now the nucleolus starts to disappear. You can see here in late prophase, the nucleolus, it starts to disappear. Nuclear membrane also disintegrates and disappears gradually. You can see here, this is the nuclear membrane. Okay, in early prophase, it was very much distinct. You can easily see, but in late prophase, you can see the nuclear membrane totally disintegrates and slowly or gradually it disappears. Centrosome which has undergone duplication during interphase begins to move towards the opposite poles of the cell. Now, as I said, centrosome is one of the cell organelles, and this centrosome, it gives rise to centrioles. Now, this centrioles, what happens? This centrosome initially it undergoes duplication during interphase. Interphase is the phase which is previous to the prophase. If we study cell cycle, we can see before cell cycle there is a phase in the cell cycle. There is a phase called as interphase, and then interphase is followed by cell division. Okay, so in interphase, what happens? Duplication of centrosome had taken place. So centrosome, it gives rise to two centrioles. Yes. So duplication had occurred. That means two centrioles were produced. Okay. So duplication of centrosome has taken place, which gives rise to centrioles. And you can see the centrioles, they are moving towards the opposite poles. Okay. And also you can see that mitotic apparatus is completely formed. You can see mitotic apparatus, it refers to spindle fibers. Mitotic apparatus, it refers to centrosome also, and it also refers to spindle fibers. So in the late prophase, you can see spindle fibers are completely formed. So this was prophase, early prophase and late prophase. You can see the difference. Okay. Next phase is metaphase. In metaphase, the chromosomes are completely condensed so that they appear very much short. In metaphase, what happens? These chromosomes, you can see these are the chromosomes, and these chromosomes, they completely undergo more and more condensation so that they become more and more thick. Condensation, as I said, that it is reorganization of chromatin material. The chromatin material arranges itself in such a way that the chromosomes, they become more short and more thick. So in metaphase, again condensation process takes place. And here you can see they appear very much short. Sister chromatids and centromere become very much prominent. Sister chromatids, these are sister chromatids 1 and 2. These are the sister chromatids. We can define sister chromatids as the chromatids of the same chromosome. This is one chromosome, this is another chromosome, yes. So when the chromatids of the same chromosomes, they are called as sister chromatids. So sister chromatids and also the centromere. Here is a centromere. If we see here, see, these are the sister chromatids. This is one chromosome. These are the sister chromatids, that is chromatids or arms of the same chromosomes. They are called sister chromatids. The two chromatids, they are folded together by a centrally placed centromere. Also, you can see kinetochore. Now, what exactly is meant by kinetochore? Kinetochore, it's a disc-shaped structure. And this disc-shaped structure, to this disc-shaped structure, you can see spindle fibers are attached. So in metaphase, you can see these are the kinetochore, which is a disc-shaped structure. And to this disc-shaped structure, you can see that spindle fibers remains attached. Now, what is the major character of metaphase? The major character is what? All the chromosomes, they lie at the equatorial plate of the cell. You can see this is the cell and this is the equator of the cell. So you can see here that all the chromosomes, they lie at the equator or equatorial plane of the cell. That is, all the cells, they arrange themselves in the equator. And this plate is called as metaphase plate, or it is also called as equatorial plate also. So all the chromosomes, they arrange themselves at the equatorial plate or metaphase plate. Mitotic spindle is fully formed. You can see in the previous, that is prophase, we had seen that centrioles, they move at the opposite poles. These are the opposite poles. This is the cell. These are the opposite poles of the cell, and this is the equator. So in metaphase, what happens? The centrioles, yes, which was produced from the centrosome. Yes, so the centrioles, they reach the opposite poles, and from the centrioles, you can see spindle fibers coming out. So in metaphase, the spindle fibers, they are completely formed. Now, what exactly are the spindle fibers? These spindle fibers, they develop from microtubules. You can see microtubules are written. So, what are microtubules? When we see microtubules, they are fragments. Okay, they are fragment-like fibers, and these fragment-like fibers, they are made of a protein called as tubulin. Okay, so this microtubule, they are fibers. Okay, they are bunch of fibers. They form a bundle of fibers, and these bundle of fibers, they are made up of a protein called as tubulin. This tubulin, they have the capacity to extend and elongate also, and that is why they have the capacity to pull the chromosomes. That we'll see in anaphase. So this was about metaphase. Also, what happens? Centromere of each chromosomes divide into two, each being associated with the chromatid. You can see centromere of each chromosome. This is the centromere of each chromosome. Okay, so this centromere, it divides into two like this. Okay, this is the centromere of the chromosome, and this centromere it divides into two, and each being associated with the chromatid. Each spindle fiber it is associated with the chromatid. Here you can see the spindle fibers. Okay, here you can see this is the centromere, and the centromere like this it divides into two. So in anaphase, what happens? From a single chromosome, you can see two daughter chromosomes are formed. That we'll see further. Okay, so centromere of each chromosome divide into two. So this is metaphase. The major character is what? Chromosomes they become more short and condensed. Sister chromatids and centromere are very much prominent. Sister chromatids and centromere are very much prominently seen. The one of the major character of metaphase is what? All the chromosomes they lie at the equatorial plate, and also centromere of each chromosome it divides into two. Next is anaphase. Now, what happens in anaphase? The chromatids of each chromosome separate and form two chromosomes called as daughter chromosomes. As in metaphase, we had seen that the centromere divides. The centromere divides, okay, and thus from each chromosome, you can see how two daughter chromosomes are formed. These are two daughter chromosomes. Yes, now, the formed chromosomes are pulled away in opposite direction by the spindle fibers. These are the centrioles, right? Okay. And from the centrioles, you can see spindle fibers. Okay. Now, these spindle fibers, as I said, it is made up of tubulin protein, and this tubulin it has the tendency to extend and elongate, and because of extension and elongation what happens, the chromosomes you can see here the daughter chromosomes are separated into two. Okay, the chromosomes in the metaphase, the centromere of the chromosome it gets divided into two, and these two chromosomes they are called as daughter chromosomes. So these spindle fibers you can see that they pull the daughter chromosomes, they are pulling the daughter chromosomes towards the opposite poles. They take on a V shape or Y shape as they are pulled to the either pole. Now if we see the shape of daughter chromosomes, they are V shape, you can see here. They are L shape also, and they are Y shape also. Chromosomes being pulled away appear like a bunch of bananas during midway of anaphase. You can see they appear just like a bunch of bananas during midway of anaphase. Now each set of chromosomes reach at the opposite poles of the cell marks the end of anaphase. Now in anaphase also there are two phases, early anaphase and late anaphase. In early anaphase, what happens? The daughter chromosomes which are formed because of division of centromere, what happens? The daughter chromosomes in early anaphase they start moving towards the opposite poles. This diagram is of early anaphase, and when this daughter chromosomes when they completely reach at the poles, that indicates the end of anaphase, that is late anaphase. Next stage, the last stage is telophase. The telophase is the final stage of karyokinesis. As I said, it is the last stage taking place in the nuclear division. Nuclear division is called as karyokinesis. The chromosomes with their centromeres at the poles begin to uncoil, lengthen, and lose their individuality. Now, in late anaphase, we had seen that the daughter chromosomes reach at the opposite poles. So when the chromosomes reach, basically daughter chromosomes, when the daughter chromosomes reach at the opposite poles, you can see this was the metaphasic plate in metaphase. Yes, and then in anaphase, all the daughter chromosomes they reach at the opposite poles. Now when they reach at the opposite poles, what happens? The chromosomes they uncoil themselves. Okay, they uncoil themselves, they become in the form of long thin thread like structure. In prophase, we had studied that the chromosomes undergo condensation so that they become short. But when they reach the opposite poles in anaphase, what happens? All the chromosomes, they uncoil themselves. Okay, they uncoil, and when they uncoil, what happens? The length of the chromosome increases. As a result, what happens? They again appear in the form of long, thin thread-like structure. The nucleolus begins to reappear. You can see the nucleolus. Here is somewhere nucleolus. So the nucleolus, it begins to reappear, and the nuclear membrane, it also appears around the chromosomes. This is the nuclear membrane. Yes. Okay, it is a covering around the protective covering around the nucleus. So in prophase, we had studied that nucleolus also disappears and nuclear membrane also disappears. But in telophase, when the daughter chromosomes, they reach the opposite poles, what happens? They uncoil, the chromosomes uncoil, the nucleolus also appears and the nuclear membrane also appears. Spindle fibers break down and get absorbed into the cytoplasm. In previous anaphase, we had seen that spindle fibers were present. But what happens to the spindle fibers when they reach telophase? The spindle fibers, they break down. All the spindle fibers, they break down and they get absorbed in the cytoplasm. Over here, this is the nucleus. Okay, so the spindle fibers, what happens? All the spindle fibers, they break down and they get absorbed into the nucleus. So hence, at the end of telophase, two daughter nuclei are formed. You can see this is one daughter nuclei, this is other daughter nuclei. So at the end of telophase, two daughter nuclei are formed. So this was all about division of nucleus, which was called as karyokinesis. Further, after division of nucleus, division of cytoplasm takes place. This is all cytoplasm of the cell. This is the nucleus of the cell. This is the cytoplasm of the cell. So after division of nucleus, that is karyokinesis, division of cytoplasm is followed, that is cytokinesis. Now when we see cytokinesis, the division of cytoplasm into two daughter cells is called as cytokinesis. You can see these are the two daughter nuclei. Okay, so after division of nuclei, what happened? Division of cytoplasm takes place. The division starts with a constriction. You can see a constriction can be seen. Okay, here also. Okay, so a constriction, the division of cytokinesis takes place with a division of constriction. Now this constriction gradually deepens and ultimately joins in the center, dividing into two daughter cells. You can see how furrow is formed. Initially, what happens? A constriction is developed. This is the constriction, and this constriction later it deepens and it forms a furrow. You can see a cleavage furrow is formed, and these cleavage furrow ultimately, when it deepens more, it divides into two daughter cells. Now, this mechanism of cytokinesis is characteristic of animal cells only. Now, this furrow, you can see this constriction, this furrow is formed. So, this mostly takes place in animal cells only. Okay, because in plant cell plate is present. We know that plant cell, it has a cell wall, but in animal cell there is no cell wall. So cleavage or furrow is formed, and by the formation of furrow or constriction, it leads to two daughter cells. So this furrow, it is major character seen in animal cell only. Now when we see cytokinesis in plant cell, this is the plant cell. Okay, so here we can see how in plant cell a cell plate is formed. So the plant cells are covered by a relatively non-flexible cell wall. Due to this, furrow cannot be formed. As I said that what happens, cell wall is present in plant cells. So plant cells are covered by relatively non-flexible cell wall. The cell wall is non-flexible, and as the cell wall is non-flexible, no furrow can be formed. Instead of furrow, what happens? Cell wall or partition starts to appear at the center of the cell and grows outward to meet the existing lateral walls. You can see initially what happens, a cell partition starts to appear. You can see a vesicle forming cell plate. So initially a cell plate is formed, a partition starts to appear. Here it is starts, you can see it is starting, okay, thus appearance of lateral walls, it is starting, okay, and then what happened, it grows outward to meet the lateral walls. These are the lateral walls. Yes, initially you can see a partition starts to appear, and this partition later it grows outward to meet the lateral walls, okay. And ultimately, what happens when the lateral wall touches the cell wall? You can see how two daughter cells are formed. So a formation of new cell wall begins with the formation of simple precursor called the cell plate that represents the middle lamella between the walls of the two adjacent cells. Okay, so as I said, here you can see the cell plate, it acts as a precursor. Okay, a simple precursor. With the help of cell plate, what happened in the middle lamella, this it acts as a precursor, and thus two daughter cells can be formed, or two adjacent cells can be formed. Now what is the significance of mitosis? What are the importance of mitosis? Mitosis, it is an equational division where the chromosome number is maintained constant. As we saw at the end that from a single cell, we had seen how from a single cell two daughter nuclei are formed. Okay, prophase, it had only a single cell, and later in telophase you can see two daughter nuclei were formed, and after cytokinesis you can see two daughter cells were formed. And in these two daughter cells, what happened? Equal number of chromosomes are maintained. You can see that in anaphase, daughter chromosomes were formed. Yes, in anaphase, what happens? Daughter chromosomes were formed. Yes, and these daughter chromosomes, they had equal number. Okay, in you can see that in daughters, that the daughter cells which are formed, they had equal number of chromosomes. So in mitosis, equational division, the chromosome number is maintained. Then equal distribution of nuclear and cytoplasmic content between the daughter cells. You can see that the equal distribution of nuclear membrane also takes place, and cytoplasmic content also is equally distributed. All the contents in the cytoplasm, basically all the different cell organelles, okay, they are also equally distributed. The hereditary material DNA is also equally distributed. We know that RNA and DNA, they are the hereditary material. Yes, that is deoxyribose nucleic acid. So here the hereditary material DNA is also equally distributed. Now it helps in growth and development of organism. Now when the cell divides from one cell, two daughter cells are formed. Yes, these two daughter cells, they will again divide to form more daughter cells. Yes, so what happens when these cells divide? They ultimately results in the growth and development of organism. Also, one of the significance of mitosis is what? Old and worn out cells are replaced through mitosis. All the cells which have become old and which had undergone wear and tear, all that are replaced by mitosis. Now, it helps in asexual reproduction of organisms and vegetative propagation in plants. Okay. Now, it helps in asexual reproduction of organisms. We know that organisms, they undergo sexual reproduction also and asexual reproduction also. So this mitosis, it helps in asexual reproduction of organisms and also vegetative propagation. Vegetative propagation means what? From any part of plant, a new plant can be developed. For example, buds. If we see potato, for example, if the bud is detached from the potato, and if that bud is what you can say, if you put it in a soil, you can see that new potato plant can be developed. So from the vegetative parts, it can be stem also, okay, and it can be leaves also, or any part of the plant. And from that single part of a plant, a new plant can be developed. So mitosis, basically helps in vegetative propagation of plants. That means there in buds and all, what happened? Mitosis takes place. And the process of mitosis, it also maintains the nucleocytoplasmic ratio. That means the ratio of nucleus is to cytoplasm, it always is maintained. Okay, so this was the significance of mitosis. I hope you understood this lecture. So today it is all for today's lecture. Thank you.